2015, students in the biology program at Mercyhurst University took up a new challenge, namely to assess the health of the marine ecosystems of Belize. Dr. Sarah Turner is the research director for this project. A partnership was struck with the University of Belize such that Mercyhurst students could use the marine research facility at Calabash Caye as their base of operations. In January of 2015, two separate teams of Mercyhurst students systematically categorized, counted, and photographed life on and near the shores and reefs of this sensitive ecosystem. The results, charted in a film to follow, sum up those findings. In 2 years, Mercyhurst will return to Belize and repeat the process. The team will then be in a position to make valid comparisons of the two data sets and draw conclusions about long-term trends. As it turns out, the trip was educative in ways we could never have foreseen.
My name is Kelsey Armstrong. I am a senior here with a major in forensic anthropology and a minor in biology. When we were in Belize, I was collecting data on hermit crabs based on their activity during the day versus the night and where they were found, like whether they were out in the open or if they were like hiding in a tree or under a leaf. I found a few areas where it seems like the hermit crabs have like the highest populations like throughout the days. And I've just been going to the same spots over the past few days and will continue tonight to determine just like the population that's there, where in the tree they are, if they're like in like a little nook in the tree, if they're in the tree trunk, if they're surrounding the tree, if they're like running away, if they're just in a random sandy place, that as well as the size of them, just to see like if larger ones go in a certain spot, smaller ones go someplace else. I can gather a lot of information just from those two data points. I found that they were most active during the night. That was when I found them most like roaming around the key, whereas during the day they all seemed to be congregating around one fern and in like little nooks in the trees, just kind of like hiding and staying maybe away from the sun. The hermit crabs are a lot more active during the night. Like I find a lot more like out and about, whereas during the day they tend to hide underneath like a fern probably for shade to keep away from the heat. The graph just shows that, well, these were the total hermit crabs that I found. The blue is during the night, whereas the orange is during the day. That's just like a total representation of the population I saw. Most of them that I found were hiding or stationary, both during the day and at night. There was definitely a difference in when, like, at the different time of the day when if they were active versus if they were not active. So there's a lot more at night that we're moving around and out and about versus at night or during the day. Homeostasis is when the body is at equilibrium, when everything is equal. Like our bodies are at homeostasis in a normal state, and if anything is disrupted, like the pH, the temperature, the amount of liquid in the cells, anything, it will react to like fix that, so it'll maintain that like constant, like through shivering, like muscle contractions, through sweating, through increased breathing, increased heart rate, stuff like that. So the increase of moving would potentially like help keep them at a normal body temperature. With climate change and how that is impacting the environment and how it is changing their environment, another thing I found was invasive species also dictate when they're more active, because if there is like a specific ant present during the day, the ant competes with them for the food. So they have to change where they're found and like where they or when they are active to find the food, so they aren't competing directly with the ants. So invasive species is a problem, as well as the different changing in temperature. There doesn't really seem to be a severe problem. The only issue is us bringing invasive species to different locations. We just have to be more careful with that and not have any more like lionfish incidents or zebra mussels. Just kind of keep them like that, because that just disrupts the entire ecosystem.
My name is Laura McCrellis. I'm a senior biology major. I was researching brain coral. I wanted to see if there was a difference in the abundance of the brain coral between the different reefs we visited, and also so that future researchers could see if the brain coral was increasing or decreasing between years. Hoping to learn what type of environment the brain coral likes, since they are a threatened species, to see if we can replicate these environments to get them to come back and get more brain corals growing. I was expecting to see more brain coral in the shallower reefs and less brain coral in the deeper reefs. I used quadrat sampling, so me and my group, we went and we had a PVC pipe square that was a square meter, and we would hold it over different sections of coral reefs, and we would count how many brain corals were in each square, so we could get an average of the number of brain coral per square meter in that coral reef. It actually showed that there wasn't any statistical difference between all 4 reefs. I believe this is because none of the reefs, even though they were slightly different depths, they weren't great enough disparity between the depths, so that's why they just were pretty much the same. Since the various coral reefs we visited looked relatively the same and were the same depth, that is why I believe my results did not show a significant difference. So if we would have visited a deeper reef, or maybe one that got more or less sunlight, then I believe my results would have been different. It's significant because a lot of fish and different aquatic species live in these corals, and the brain corals as well. So without these types of corals, the fish will no longer have places to live, and so not only will the coral species be dying off, but the fish as well. Well, hopefully future students or researchers can go to these areas and also research the brain coral. This will help us to see if this type of coral is decreasing, increasing, or staying the same in these areas, which can help us tell if the fish populations will decrease or increase as well. Yes, so this is my graph, and on the x-axis I have the various coral locations that we visited, and on the y-axis I have the number of brain coral per square meter. So each one shows the number above the chart shows how many brain corals per square meter. So they range from about a half a brain coral to 1 1/4 brain coral per square meter. We definitely need to be more careful with, recycling and different things like that, because that all plays an effect on the coral reefs. And while I was there, I did notice a ton of coral was bleached, which means that that's dead coral, which is not a good sign. So not just brain coral, but all coral in general. So by definitely watching, what plastics we're using and recycling and all, using less water, that kind of stuff will definitely help our coral reefs.
Hi, I'm Sam DeSalle. I'm a junior here at the Hearst, and I'm a biology major and a chem minor.
I was looking at the biodiversity of fish around the reefs of the Calabash Caye where we were staying, and I was trying to determine the biodiversity through a time swim. So I'd count the different number of species to kind of see what the overall health of the system was. So time swim, what we did for ours was we sampled 4 reefs around Calabash Caye, and from there we picked like 20 spots, which then each of the reefs, and would hover over one specific spot of those 20 spots, and for 2 minutes count the different types of fish that swam by, making sure we only counted the species once. So if like 20 of the same fish swam by, it was only technically counted as once. And we recorded all the data, did it 20 times for each location, and we had 4 data sets.
Biodiversity is the number of different species, whether it's plants, animals, insects, that's an animal, but just any type of living organism, the different types there are. So the more you have, the more diversity you have.
Going in, I kind of reasoned that I would not see a difference in the biodiversity from the 4 reefs just because of their close proximity. The water is pretty much the same. As we got further away from the Caye, it got a little bit more salty, but I didn't think that'd have a huge impact on anything. So I was expecting my numbers to sort of be the same for the average biodiversity from location to location. Okay, so my results showed I had fish anywhere from 3 different species to about 20 different species from place to place, so the numbers were kind of varied. But all the averages from location to location were about the same, which is about anywhere from 10 to 12. So my hypothesis was supported by the data I collected, and there wasn't really a significant difference in the biodiversity of fish from reef to reef. Okay, so the conclusion was that my hypothesis was supported. You're going to need more data to really truly support it, because only 4 reefs were sampled and only a small number of data sets were collected. But preliminarily, that does seem to be supportive. But I mean, there's also a lot of factors you have to consider. Like a large group of people were constantly swimming in the area, so that can drive your numbers down or kind of keep the numbers the same. When you have people, a lot of swimmers in the same area, you have people swimming through in the middle of your time swims can kind of drive your numbers down. But because all of those factors were sort of present in each of the 4 locations, you saw sort of similar results. So what we did was, at each of the 4 locations, we got in the water and we worked in a group of 4, so 2 at a time. We do a time swim for 2 minutes, did that 20 times, recorded all of our data, and when I got back, I ran an ANOVA test, which is just a sort of statistical analysis to determine if there was a significant difference between the 4 locations and the biodiversity that we're seeing, and it was determined that there was not. Biodiversity is really important in determining how healthy an ecosystem is. If you see a large number of different species, it can kind of indicate that, hey, this is pretty healthy, it's able to support life. But also, it's important to look at the specific species that you're seeing. So when you see a lot of the parrotfish, which are the algae eaters, it's showing that the coral itself is healthy. But then you also see, we saw lionfish, which are the invasive species that don't have a lot of the known predators besides the grouper, which have been overfished. So just because you see a lot of fish, it's important to determine what type of fish you're seeing. So, for example, yeah, we have the parrotfish, which are good, but you also have those lionfish that are going to eat a lot of the native fish and kind of drive numbers in weird ways. Looking at biodiversity, it's important to know, like, hey, let's not overfish, let's try and keep these numbers as they are, help improve them, maybe reintroduce species that are no longer present in the area but once were due to overfishing or whatever reason. Looking to see the types of fish, like if you do have a lot of the lionfish, you kind of want to get those out because those are that invasive species that aren't good to have in your ecosystem. So you want to get those fish out. And it's just important to know that, like, we shouldn't disturb the ecosystem. So knowing what's there kind of gives you that appreciation value that this is what's here, let's not ruin it.
My name's Carly Blanding. I'm a junior biology major.
My research was on the mangroves and the sponges, on the red mangroves and the symbiotic relationship between them. And we went to 2 different mangrove areas. The first one was a little rough because it was covered in jellyfish, so you had to be really careful swimming through it. And then the second area we went to, I didn't get as much data, but it was a lot easier to swim through there because it was deeper.
I was expecting to see more sponges on the roots that were healthier because that would, if their relationship is good, they have more of them. So I was expecting to see more where would get more sunlight, the roots would be able to grow easier. I did a transect with my group. We all went in the water and we went to the roots. We're each a meter apart and we counted all of the sponges that we saw in our area, and then we would swim for 10 seconds so we were all in a new place and do it again 1 meter apart. My results showed that there were more sponges inland and less in the open water, which was a little different than I expected because I figured that there'd be more sunlight in the open water because it'd be shaded where it's inland, but that's where they seem to be healthiest. I think that I would have to go back and do more research to really figure out the cause of why there's more sponges inland, so I'm really not sure the reason behind it. We could learn more about the relationship between the sponges and the mangrove roots and what makes them grow. I think just the fact that there's less sponges in the open water, then maybe there's something there causing that we could look into and change for the future.
My name is Mary Barnes. I'm a junior here at Mercyhurst. I'm majoring in biology and minoring in genetic counseling. My research question is, what is the distribution of different sea sponges on the red mangroves? There is a mutualistic relationship between the sponges and the red mangroves. The sponges help to sequester carbon and nitrogen for both the sponges and the mangroves to use. Obviously, you need carbon, nitrogen, those kinds of things for cellular activity, for photosynthesis, to maintain life. My hypothesis was that there would be more sea sponges on red mangroves that were in good health, that meant they had a lot of sunlight, the pH of the water around them was about 7, that they had a good enough amount of sedimentation around them that they were able to grow, and hopefully they were both of equal abundance. I collected research fairly simply. We had a quadrat, it was a meter square round, and we took it and we placed it right at the tip of the roots, at the tip of the water that the mangrove was at, and we just counted the number of sponges. There were orange ones, blue ones, green ones, brown ones, so you had to collect them all. And then we traveled, I think, every third mangrove and measured again. That way we kept it about as even as we possibly could. We took about 25 quadrat samples at each site. My results showed that the current mangrove, which was off of the coast of the Calabash Reef, that one had a much higher abundance of sea sponges as opposed to the jellyfish mangrove, which was kind of on the back behind the main water. So it's a little bit more shady than the current mangrove was. That could have been the reason why there were not as many sponges found there. They use sponges as like have you ever heard of the canary in a coal mine aspect of when you're measuring health? They say that if a mangrove is healthy, then you're going to find more sponges. And if you aren't seeing a lot of sponges, it's indicative of the health of the mangrove. At this point, no. Had I measured something like the salinity of the water, the pH of the water, the sedimentation, I could have gotten a more solid result as to pinpoint exactly why there were more sponges on one reef than the other. But I just simply measured. It was just a simple beginning experiment as to the abundance of the sea sponges. Well, it helps you if you're trying to find the health of the mangrove, especially because of how important they are to the ecosystems. You see that there aren't a lot of sponges there, you should automatically think that there is a problem. There's not enough sunlight, there's not enough, the pH isn't correct. There might be too much trash littering it, so around the area around it, so they aren't as healthy, they aren't able to get clean water. So when you are trying to look at, what's wrong with the ecosystem, how healthy is the ecosystem around this area, if you don't see a lot of sea sponges, you know that there is something wrong with the water. Now that we have the knowledge of the sea sponge abundance on the mangroves, you're able to look at maybe, there's too much litter because we know that's a problem. Red mangroves act as a natural water filter, so maybe with all the increased pollution there, that the mangroves, there aren't enough mangroves to go around, I guess, to filter the water effectively, and so the remaining mangroves actually are suffering from this. So making sure that pollution is being kept to a minimum is really huge. If we were to have another class go back to Belize, they could further my preliminary research. They can test the same mangroves that I studied, and they can see if the pH is the problem, if litter is the problem, if sedimentation, pH, any of that stuff is the problem. They could be studying those things, and that could further help scientists to understand what's going on with the mangroves right now.
My name is Hannah Green. I'm a junior at Mercyhurst, and I'm a biology major. I'm studying coral bleaching, and I'm trying to see if there's a pattern, whether they're more bleached if they're closer to the shore or further away. So coral bleaching, coral are an animal, and the way that they survive is they have a symbiotic relationship with zooxanthellae, which is a photosynthetic algae that uses light to produce glucose. So the coral use the glucose that's produced by the algae, and that's how they create their calcium carbonate skeletons, which is basically their structure. So when coral live in a very specific environment, and when the environment changes, or especially if the water gets too warm, or there are changes in water pH or salinity or turbidity of the water, the zooxanthellae will leave, and this gives the coral this white bleached color. So without the zooxanthellae, they don't have any
source of glucose, and then they'll die. Or they can recover if the environment returns to normal range.
My helpers and I we take a quadrat and we go out and we try to find a piece of coral that's shallow enough that we can get to it. And we take the quadrat and put it over the coral and we take a picture. And then later I'll go back and count how much of how many corals in the quadrat are bleached.
Basically, when you do a biological study, you're trying to reject a null hypothesis. So the null hypothesis means that there is no significant difference. Any biological study, you have to use statistical tests in order to prove if there's a significant difference, or else you don't really, you're just looking at stuff. So I used an ANOVA test, and for an ANOVA test, in order for your difference to be significant and to reject the null hypothesis, you have to get a p-value above or below 0.05. Reefs are obviously bleaching at a very high rate, and that was one of the things I did find in my study, was that it didn't matter where we were really, there was a lot of bleaching. And it's caused by rise in temperature of our oceans and overexploration and change in pH and such. So reducing pollution, reducing your carbon footprint, and exploring less, which is kind of hypocritical because we were exploring the reefs to do the study. But it really does harm the coral when, you know, people are bumping into them, or it's being touched or broken. So I guess careful exploration would be should be emphasized more in coral reef vacations.
Hi, my name is Maggie Matthews. I'm a public health and biology major at Mercyhurst University, and I'm a senior. My project for my research when we went to Belize was that I was looking at the diversity of fish that lived in mangroves. Okay, so mangrove is a tree. We've been looking specifically at the red mangroves that have its roots that grow down into the water off like the coastline. So it's just a tree. I'm using a timed swim, and that means that we're going 5 meters in between, and we time it for 30 seconds and count the different species of fish that we see. I was hoping to see like a large diversity of fish within the mangroves to try and like stress the importance of mangroves on the ecosystem. So my results were that we went to 2 different locations with the mangroves. The first location, there was more of a current, it was deeper, there was more visibility. And the second location, it was shallower, there was a lot more seagrass, and the visibility was lessened. So I compared between the 2 mangroves where there was a higher diversity of fish, and I ended up with the results of there being more diversity of fish where the water was shallower, there was less visibility, and there was more seagrass. In both locations, there was quite a diversity of fish, which goes to show that the mangroves are a great part of our ecosystem and that a lot of fish need it as a shelter and a home, so that we should reduce the amount of deforestation of the mangrove trees and reduce how much we're cutting the mangroves down. My results were significant because they show how important the mangroves are to the ecosystem, to humans, to fish, pretty much to the entire aquatic environment along with the land. It's important because we want to know like how much impact it is making when mangroves are removed from the coastline. So not just like the coastline or the issues that we'll have with the coastline, because like the mangrove trees provide a buffer for the coastline, so but also what's happening to the aquatic environment underneath. Yes, this knowledge does improve what we know about the mangroves. We do know how important they really are to the ecosystem, and we should really take that into effect and account when we do what we do. Humans destroy a lot in our path because we're more worried about us than everything else, but we don't realize how much we impact ourselves when we destroy things like mangroves, because it's going to lead to erosion of the shorelines, which is going to reduce the amount of land we have to live.
My name is Brianna Flynn. I'm a junior anthropology major bio minor. I looked at the distribution of the mangrove species as it went along with the salinity of the water. So I took samples out in the ocean, and then I took samples more inland to see how the species differentiation changed red to white. I expected to see more of the red mangrove species closer to the ocean, so the more saline water would have more species of red mangrove trees as opposed to black and white. I used a transect sampling method, so I would pick a point on the shore and then just take salinity samples as you go out into the ocean and then back inland in a straight line. And I did that in a couple of places on the shore. It was pretty consistent with my hypothesis. As you went closer to the water, you saw more red mangrove species, and as you got further inland and the water became less saline and more freshwater, then you would see first black mangroves and then white when you got to a point where there wasn't any water on the surface of the ground. I would definitely find a better way to take these salinity samples, because as I did it there, you had to use like the pH strips, and swimming with those across like large bodies of water was really risky because you could have contaminated samples. So there is a possibility that some of my data was messed up because the pH strips couldn't stay dry. Also, maybe find a place where there were more species diversity, because as it was on the Key, there wasn't a lot of white mangrove species. It was mostly red and black. So seeing some white mangroves would probably be a little nicer. Well, red mangroves are the kind of stereotypical mangroves that people think of with the prop roots, and they live in deep water. The black mangroves are a little more bush-like, and they have projecting roots out of the ground, like they go down and then they come back up. And that's kind of how you differentiate them. And then white mangroves just look like regular trees that you see every day. They have kind of a speckled bark on them, and the really big distinction of mangrove trees is that they can live in freshwater, brackish water, or salt water. And so they have different adaptation to filter the salt out so that they can actually use the water.
My name is Dan Karstead. I'm a junior biology major. My research was counting the abundance of sergeant major fish in the coral reefs off Belize. My hypothesis was basically that each reef that we visited would have a different abundance of the fish that we're looking for. The method we used were transect sampling, which we start on the boat and then you get in and you start at 0 meters. You count from that location, and then you would swim another 5 meters and then count from there, and you do that all the way up to 50 meters, and then you swim a little ways this way. We did 20 meters in between each, and then swim a parallel line back towards the boat. And then you just keep doing that until you get enough of your sample. We used methods called transect sampling, which is when you get off the boat and then you swim for about 10 meters and then count from that spot, and then you go on to the next 10 meters. And you do that for about 50 meters and then go to about 10 meters away and then do the same thing going back just going in parallel lines, counting fish at each spot. We counted a total of 620 sergeant majors in all of the reefs combined. One reef had 159, and then a couple only had 11 or 12. So it definitely showed a difference among the reefs. The conclusion we can draw from that is that each reef has different characteristics that are better or worse for the species. One of the characteristics is possibly the water depth and the depth of the coral, because they look like, from my research, I found that about 1 to 12 meters is the best depth for sergeant majors to live at. This is very preliminary, and we'll need to do more research on it, on the coral themselves and maybe the predators of the sergeant majors. And in the end, hopefully we can look at the health of the reef ecosystem in Belize and assess what we need to do to change things down there. I would probably look more at the coral and how tall the coral are, how big the coral are, what type of corals they live in, and how close they are to the water to see how the habitat itself affects the abundance. I think if we just went to more reefs and got a larger, able to get larger sample sizes from different places. So this graph shows we did 14 total transects, and it was over 4 days, so 3 different reefs that we saw. And each line is just vertical here, and that's how many we saw at each location. And you can see it range from 11 here, and then one of them we saw 159. It was a pretty wide range, different at each location. And the total was 620.
I'm Kelly France. I'm a junior biology major. My research was looking at coral bleaching and seeing the frequency of coral bleaching in that area. My hypothesis was that the frequency of coral bleaching would be less when the corals were closer to shore as opposed to further from shore, because I thought that the corals closer to shore would have more evolutionary adaptation to different stressors. In order to do the research, I used quadrats, and we just would set like a 16 square quadrat over the coral and just count how many of the squares coral bleaching was in, and then we just swim 20 seconds between each location to get different data points. It's just, a quadrat is just like a square that has 16 different little squares in it. So you can just use that to find the frequency. Okay. The results varied a lot. I ended up not finding a significant difference between the coral closer to the shore and the coral further from shore, because there was just such variability. Like some had 0, some had 10 out of 16 of the squares. It just completely depended on where the quadrat was. I wasn't able to find enough data within the time allotted to be able to draw a sufficient conclusion. I think this is definitely a big area of research because global warming has a major effect on coral bleaching, and corals are very important. So it would be good to do further research on it to see what effect it's having on different species within that ecosystem. Coral bleaching is when coral lose the symbiotic relationship between them and algae. And the algae is what causes them to be pigmented. So when they lose this relationship due to various stressors, they become bleached or white. We should definitely consider things like CO2 emissions and stuff like that. Just seeing the effects that what we do have on the environment is definitely very important, and like realize what we are doing to all different types of ecosystems. If I did this again, I would just like more time to spend in the water and really be able to look at them and do more research before to really understand what I'm looking at and the species of fish and things like that.
My name is Megan Polakowski. I am a sophomore here at Mercyhurst University, and I'm a biology major. My project was based on the coral reefs that we found along Calabash Caye, and I went in and studied the diversity of fish based on how deep the corals were into the ocean. My hypothesis was that the more shallow reefs were going to have a larger diversity of fish than the deeper ones. Well, first we had a group of us circle a certain piece of coral in the area. We then took measurements from the sea surface down to the topmost piece of hard coral that we could find, and then we counted the fish that were around the coral reef that we picked. My results show that there was a correlation between the depth of the coral and the diversity of fish that was found in that piece of coral. So with the shallower corals, we found that there was a lot of fish swimming through them, and the deeper corals, there were just smaller amounts of species but larger fish there. Well, I think it would be great, since we have a lot of coral bleaching going around the world right now. It'd be interesting to see how planting new corals deeper versus shallower in the ocean would actually help the corals grow faster compared to slower by how the fish actually accumulate to that area.
My name is Kelsey Johnson. I'm a junior biology major. I did research on the distribution of coral colors within the coral reef, so like comparing the colors that were seen on the edge of the coral to the center of the coral reef. My hypothesis was that there would be more diverse colors in the center of the reef, where there would be more, nutrients for them to grow, more fish around them, and then as you kind of moved out, there'd be less diversity of color. My results showed that my hypothesis was actually not supported, so there was no difference in the diversity of color based on where in the reef they were. What we did is we worked in a team of about 5 people. Each day we went out to the reef and we would swim for 20 seconds, take a measurement. So you would use a quadrat, and you would hold the quadrat right under the water, and you would count the color of the corals. So like if you saw 4 purples and 3 pinks, you would write that down on a little board, and then you would swim another 20 seconds and just repeat all the way from the very, very edge of the reef into the very center. The conclusion that we got is really that the color of coral doesn't depend on where in the reef it is. It depends on the symbiotic relationship of different types of algae and other things like that that react with the hard skeleton of the coral itself. And so there's no difference in the edge compared to the center. The color of coral and the different types of colors, so the amount of different colors of coral in a coral reef, show the health of coral reef systems. So when you see a high diversity of color, it means the coral reef is healthy. So seeing a large diversity everywhere is a really good thing. So when you're looking for the diversity of coral color, I guess you're just looking to see the health of it. Even just while I was looking for the specific colors, there was another girl who was researching coral bleaching, and my project was affected by coral bleaching a lot too. So as many times as we would see diversity of color, we would see just as many bleached corals. And coral bleaching happens from environmental changes. So whether there's chemical imbalances in the water, there's trash, there's more or less fish than the coral needs, the symbiotic relationship is disturbed. So when you're seeing significantly bleached coral systems, it's something that humans are causing or humans are contributing to, and it's something that we need to really pay attention to. I feel like visiting a more diverse amount of reefs, so a lot of times you would spend an entire day in one reef and then we'd come back there the next day. So I'd be collecting, 2 of the same data sets, and so having different types of coral reefs as well as more time at them to collect the data would probably be helpful.
Hi, I'm Kylie Lurch. I am going to be done in May, and I am a biology major. I was focusing on trash, and as you could see, like as soon as you went there, the coastline was filled with trash. So I was trying to figure out like where the trash like seems to go according to like tides and like the size of trash, because you get like a ton of different kinds. So like if they're small pieces or like very large pieces, and I was just trying to figure out if there was any trends with how it like goes onto the shore. So we had 4 zones for the like the coastline. There was like the sand by like the shore, and it was like completely clean. It was all sand. And then the second zone was just like the seagrass, and it was just like very thick with like dense grass, and there was trash in there. And then the third zone had seagrass and sand, so it was like a mix of the two. And then above that was just sand, and then the trash that was on there. And my hypothesis was that depending on the 4 zones, the trash would change, like the size of the trash would change, and like the amount would change depending on the zone. We like broke them into teams of 2, and we would go down like each zone, and we had like the grid, the quadrat, and we would like just like drop it down, and like wherever you dropped it down, you would like count the pieces of trash that there were, and then we would like split them into small and like medium and large, and just like take note of like which sizes were which, and then you would go up on the same line of the zone, and you would like keep going into each zone until you were done, and then you walked 10 feet, and then you would do it again. My results showed that the seagrass had the most, and then the farthest up part had the most small pieces of trash, and then the zone by the shoreline was completely clean because I think the ocean just like pushed it off, and like it would just go back into the ocean. And so like there was definitely a difference in sizes, and I had a graph that showed like the difference in sizes, and it seemed that it like would increase the farther you went in to the coast, but it didn't show that there was like any like huge like difference between each zone. But like you could see that there was, but I think there was just so much trash that it like didn't make a difference, and I think we would have to do more of the same thing to like try to like see if there's trends that we can actually like test for. I think that if we would do more tests and we had like more people to do it, that you would see that trash like goes up like further on the coastline because of the tides and stuff. And if there's like seagrass and stuff, that kind of like creates a barrier. And like the bigger trash gets stuck in like the seagrass because like that just like gets caught in it, and then the little tiny pieces like get like further up onto the sand. It's significant because every coastline right now basically has trash, like no matter like where you go, and it's because like our seas are so full of trash, and a lot of people don't like really realize that because of the tides. Tides will like circle around, so like trash will just float out there for like years, and then if there's like a storm or something, it will just like float up like on a nice coastline. So people who go on like vacations are just like there's so much trash. And then for birds tend to eat the trash, and then they usually die. So it's like a very bad thing for humans and for birds and just for anything, like basically. I think people should be more aware of what they do with their trash. They shouldn't just like throw it away or like throw it on the ground. Or I have people were so shocked by like we found shoes and like toothpaste and like toothbrushes, and it's just crazy to think that like we don't take care of like the world that we like are supposed to be in, and we should take more care of it and like be sure that we do our part and not throw away stuff, or just like be like more aware of like what we do. I feel like there's a ton of like other experiments out there that were like mine. I just didn't have like the amount of time people have had or like the numbers of people to help me. So I think that mine is like a start to like what people are doing right now to try and like help an environment. But I just think it's like really important that we like get people out there to like try and like clean up the trash as much as we can.
After our 2 weeks spent researching and exploring the fragile ecosystems of Calabash Caye, we have gathered a lot of valuable information on the coral, native fish, and other marine life. We are hoping that in 2 years we will be able to return with the intention of furthering this research and our knowledge. We expect to highlight the significance of human action on these ecosystems, so we may one day reverse any negative impacts and preserve nature's splendor. Through this video and our research, we are providing education and knowledge for everyone to see the effects we are having on our Earth.